So what are some of the genes that can go wrong in cancer? There are two main classes, and here's what I want you to find in these breast cancer data sets. One are called oncogenes. These are like your criminals, right? You also have tumor suppressors, and I think we've kind of talked about it. These are your cops. If your criminals get too powerful and overtake your cops, then you have cancer. If your tumor suppressors are too weak to take on the oncogenes, which are the criminals, then you get cancer. There's a balance between these two. The tumor suppressors are always supposed to win. But here's what happens is you have these mutations where something bad happens. Guys, can you please listen, Balboa? Okay. So what are some of the oncogenes? What are things that would cause a cell to divide uncontrollably? I mean, they're given here. Growth factors. Signaling molecules. Say you tell the cell, you just keep telling the cell, grow, 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 grow. And this tumor suppressor isn't there to tell the cell to stop. It's going to keep growing. Cell cycle genes, obviously. Everybody is familiar with mitosis? Yeah. Remember mitosis? Was it IPMAT or what is it? Interphase, prophase, metaphase, anaphase, telophase. Yeah. Those genes associated with that a lot of times will get mutated. Angiogenesis. Does anybody know what angiogenesis is? That is the growth of blood vessels. So here's what also has to happen with tumors is that you form this big mass of cells, it has to get fed. If there's no blood supply, the tumor will die. So what it does is it actually produces all these signaling molecules to make the blood vessels go to it. It basically gets itself lunch. Telomeres. Anybody know what a telomere is? Any idea?
Is it like telophase or like the centuries?
No. They are kind of related to centromeres, right? So centromeres are that point in the middle,
yeah,
the chromosome. Where do you think the telomeres are?
Ends.
Ends, yes. Here's what happens. So your body has this natural way to control for cancer, is that every time your cell divides, a little bit of the end of your chromosome gets lost. The more times it divides, and basically what your cell does is put a little cap on the end of your chromosomes. Once that cap is gone, you basically start getting into your genes and then you kill the cell. What happens in some types of cancers, they keep reproducing that cap. So normally that you only have that cap and it doesn't grow at all in your life. And in fact, some people believe that if you can measure a person's telomeres, you can predict how long they're going to live. So that's kind of cool. Everybody's like, what? Yeah, I'm going to be able to see the length of my telomere this summer. It's going to be so cool. So what happens is basically you kind of have a fixed telomere. In cancer, you'll mutate a gene that will keep growing that cap. And so these cells can grow over and over. It's almost like getting invaded. Tumor suppressors, a lot of cell cycle inhibitors, DNA repair enzymes are big on this. You have a lot of genes in your cell, in your genome, that kill your cell. That you are a multicellular organism. That losing a couple cells is not a big deal. And in fact, there's a whole program called apoptosis. Has anybody heard of apoptosis? All right, Balboa, you got to remember apoptosis. I will ask you that repeatedly to the end of the semester. What is apoptosis? It's programmed cell death. Apoptosis, programmed cell death. That is basically your cell committing suicide. What happens, though, is you lose those things. So some of the big tumor suppressors are P53, BRCA1, retinoblastoma 1, and this P10. And we're going to see this a lot in breast cancer. Remember, when we were just kind of playing around with breast cancer, we saw P53 mutated and all those things, right? Totally. P53 is the big cop. And it basically sits over and makes sure that your DNA... Yes. It'll come in and just shut everything down if anything's screwy. What happens in lots and lots of cancers, you have to mutate that. So not only do you see P53 mutated in breast cancer, you see it in lung cancer, bladder cancer, lots and lots of cancers. So here are some of the hallmarks of cancer. You have it's able to give itself signals to grow. There's an insensitivity to anti-growth signals. So basically the cancer ignores the COPs. It has the ability to basically leave the tissue where it was formed and spread. Limitless replication potential. So there's this book out there. There's this famous cell line. It's called the Hela cell line. And people have been working with a cell. So basically, you could take a cell from a human, put it in culture, and it'll grow in an incubator, and you could just keep growing it forever. They got this cell line. This one woman had this, what was it? It was an ovarian cancer or cervical cancer. Her name was Hela. It was so bad that they cultured the cells that she had, basically took the cells from this very aggressive cancer, put them in a plate, and these things would grow on its own. That's how severe the cancer was. So ever since then, we basically have been using this woman's cancer to do all these like cancer experiments. She totally died. And that was the thing is, until they wrote the book, no one knew who she was. She just kind of died no one knew until finally they told the story about, her sacrifice probably cured lots of different diseases because we could use her cells. Invading apoptosis, sustained angiogenesis. Okay. And what time is it? Oh, 3:05. Okay. So here's what's going to happen is I'm going to tell you about next class period, we're going to talk about lung dysplasia, lung tumors. I'm going to tell you the thing that I found in that, and then we're going to go over basically what I want you to look for in breast cancer. So until then, just start reading some things about breast cancer. What are some of the genes associated with it? What does it cause? What are the stages? Do your own homework so next time when I start talking about breast cancer, you'll be like, I get it. All right. We'll see you on Wednesday. And hopefully there won't be another stupid... Yeah.